So DNA replication is the process of making copies of the DNA. Really, really important so that our bodies can go through mitosis and even meiosis. Remember the cell cycle. Okay, this is where the cell spends most of its time. And in the S phase is where we're going to copy the DNA. This all happens in the nucleus because remember, the DNA never leaves the nucleus. During the S phase, we go from 46 chromosomes to 92 so that we can break it up during mitosis back into 46. So we're going to draw the backbones a little differently in this video. Instead of drawing the sugars and phosphates like we normally do, we're going to draw them as just lines. It will simplify pictures and it'll make understanding this process a little bit easier. So semi-conservative replication, this is the process of actually replicating the DNA. If you see on the left, we have two blue strands. Both halves of the helix are parent strands. But when we make copies, we end up with only one half parent and the other half is new or daughter strands. The first enzyme involved in opening up and copying the DNA is an enzyme called helicase. Helicase is like an unzipper. It literally goes through and breaks the hydrogen bonds. So helicase breaks the hydrogen bonds. Now, once helicase does its job and opens up that DNA, those bases still want to connect. They're like magnets. So the helicase comes along, opens up those hydrogen bonds, but the complementary bases still need to be held apart. These are kept apart by single-stranded binding proteins. Very important to keep the DNA open.
So now that the DNA is open and helicase has done its job, we need the new enzyme. The next enzyme involved is DNA polymerase. This is the builder, or what we call the matchmaker. And what it does is it finds free nucleotides that we're drawing here. These are just floating in the nucleus. So DNA polymerase finds the correct complementary base nucleotide and just continues to build the DNA to match the bases. So we'll end up with two strands of final DNA when we're done. Each DNA will have a parent strand and a new strand. This is semi-conservative replication. DNA polymerase, however, can only read the original DNA from the 3' to the 5' end. So on the top part of this DNA, it's easy to read because it just follows the helicase from right to left. That's the leading strand. But on the opposite side, or the bottom side, this is our lagging strand because DNA polymerase has to work opposite of helicase. So DNA polymerase needs a start signal, or what we call RNA primase. RNA primase is going to tell the DNA polymerase where to start. Now on the leading strand, you only need one of these primases. And then DNA polymerase will just work down the strand and follow helicase. On the lagging strand, however, on the bottom strand in this case, we are going to need several RNA primases. We have to work in segments because remember, the DNA polymerase is working in the opposite direction of helicase. These segments that we create on the lagging strand are called Okazaki fragments. And in the very end, these fragments need to be glued together, and all the nucleotides need to be glued together. That's the final enzyme involved, which is what we call ligase. It's like the glue. So this is just a list of all the steps. Remember, helicase is our unzipper. SSB proteins are going to hold that DNA open while we do the rest of the work. Primase is going to be our initial start signal to tell DNA polymerase where to start. DNA polymerase will build from 3' to 5', and then the ligase will glue it all together.